Hello, everyone, and thank you for joining us today. During today's talk, attendee will be muted as usual. If you have any question at any point during the presentation, you may submit it in the chat panel. At the end of the talk, there will be a Q&A session. If you'd like to ask your question yourself, let us know and we will unmute you. And also remember, there is a dedicated Slack channel for this talk on the BSIS workspace. And also as a reminder, this presentation is being recorded and will be shared publicly. Okay, so it is my great pleasure to introduce to you a distinguished scholar and researcher, Dr. Sepide Sedqiani. Dr. Sedqiani is an associate professor of psychology and bioengineering at the University of Illinois, Urbana-Champaign. She directs the Connect Lab at the Beckman Institute for Advanced Science and Technology, where she is a full-time faculty. Dr. Sedqiani received her PhD in neural and behavioral science from Max Planck Graduate School and postdoctoral training at Berkeley and Stanford. She investigated the role of neural connectivity and spontaneous brain activity in cognitive control and behavior through a multimodal lens. She also served as a handling editor for Imaging Neuroscience and Network Science. Dr. Sedqiani has been recognized as a National Science Foundation Career Awardee, and her work has been funded by both NIH and NSF. Dr. Sedqiani, thank you for being here, and without further ado, I will let you take it from here. Thank you.
Thanks so much for that kind introduction. It's great to be here. Thanks a lot for the invitation. I'm looking forward to our discussions. All right, so I'll be talking about what I have come to call the connectome multiplex, and I hope it makes sense by the end of the talk why I'm choosing that term. So just to make sure I get to it and I don't run out of time, I'll start with the acknowledgements, because a lot of the data that you will see today was actually shared with us graciously by other groups. This includes concurrent intracranial EEG fMRI, scalp EEG fMRI from other groups, scalp EEG in a large data set, as well as genetics neuroimaging with collaborations on the HCP data set. And of course, all the lab members who are actually doing the work. So the talk has actually 3 sections. So I'll start with the slow connectome, which is the one we know in terms of function connectivity we know best, which is the story told by fMRI. I'll move then to the fast connectome with more real-time methods, and then the combination of them, which results in this viewpoint of the connectome multiplex. So just as a general intro, what we know from fMRI is based on the slow hemodynamic signal, the slow connectome dynamics. So let me just give you a few pointers here for those who are less familiar. The functional connectome is mainly carried by spontaneous fluctuation in intrinsic brain activity. And by intrinsic, I mean anything that is not directly evoked by external stimuli or external cognitive challenges. And so when we look at brain activity with fMRI, we see spontaneous fluctuations. The signal goes up and down. And between certain brain regions, this co-fluctuation may actually be pretty strong, whereas other sets of brain regions may have less temporal dependency. If we calculate these temporal dependencies, then we see that even in the absence of any task, or really during any mental state, including cognitively demanding tasks as well as absence of tasks, we find these neurocognitive networks that spontaneously co-fluctuate, these very well-defined connectome patterns. The first one was discovered in the somatomotor network, but it exists in early sensory areas like the auditory network, the visual network, as well as higher order networks that we actually first knew from neuroimaging and task-based activations, such as the dorsal attention network, and really the most canonical sets of neurocognitive networks that we know from task-based co-activations. So if we calculate these kind of connectivity values across all possible region pairs for a parcellation of choice, we see this arrangement with high within-network connectivity in these canonical networks. And so this is essentially a matrix representation of the functional connectome. But as we've come to realize, and your lab's work has majorly contributed to this area, is that the connectome organization changes ever so slightly over time, over the recording period. So it's not really a fully static arrangement. So these are, again, the canonical neurocognitive networks. And in this case, we had a data-driven approach to find recurrent connectome states, four states, and we replicated with other numbers of states. But if you just look at the arrangement, it might be a little hard to see, but for example, if you focus on the visual network, you see that in certain states there is higher within network connectivity than in other states, and similarly, there might be high between network connectivity, let's say between visual and the dorsal attention in certain states rather than others. So both within and between network activity, connectivity or coactivations are fluctuating over time. If you model these recurrent states, you get temporal characteristics such as the overall time spent in each one of these states over the total recording time, which is called fractional occupancy, as well as the probabilities of transitioning between any pair of brain regions, which is not random. It is very trait-like for individuals, which I will come to in a moment. So what we know about slow connectome dynamics, as I just introduced them from fMRI, is that they affect performance or cognitive outcomes from one moment to the next within the same individual. So for example, if you look here, this is the brain connectome represented now in graph space for one participant. And in this task, we gave the participant a very faint stimulus, a very faint sound, and the participant pressed the button whenever they heard the sound. And you can see that the connectome right before successfully hearing the sound and reporting it looks quite different from the connectome state prior to missing the sound. And so in this case, the neurocognitive networks here represented by different colors are more segregated. We say the brain is in a more modular state than it is prior to missing the stimulus. But there are a lot of studies that also show that connectome dynamics are associated with cognitive abilities across individuals. There are a lot more studies than this in various different kinds of populations. But what's also important is that these cognitively relevant connectome dynamics are highly heritable. And so heritability is typically assessed in traditional twin studies where twins that are genetically identical are compared to dizygotic twins. And these are my twins about maybe 4 years ago or so, and you can just eyeball and see they're quite different from each other, quite as different as you can be. So as I mentioned, if we look at these statistical properties of the temporal organization of connectome states, they are individually quite specific. So this is one participant from the Human Connectome Project data set, and you can see that this participant spends quite a lot of time in brain state 3, for example, and very little time in state 4. You can also look at the probabilities to transitioning between these different states, and some transitions are much more prevalent than other transitions. So now if you look at the identical twin of this person, you can just visually appreciate how similar their fractional occupancies are, as well as their transition probabilities. And here on the other side, I'm just plotting two different participants, which are genetically unrelated to each other, with vastly different statistical properties, temporal properties of connectome transitions. So if we look at that now in the larger population, so a larger group of subjects, this is again the full connectome data set, and we compare identical twins with dizygotic twins, non-twin siblings, and unrelated participants, you see a strong impact of genetics on the similarity, or rather dissimilarity as plotted here, of the fractional occupancies, so the time spent in the different brain states, as well as the way they're transitioning into each other. We used variance component modeling to estimate the amount of variance in these two properties explained by genetic effects, and it's about 40%, which is quite strong. And also, when we look at cognitive associations of the same heritable features, we find that they actually explain certain amount of variability in the cognitive abilities of these participants. I should also point that we have looked now, because of the heritability that we found, we've looked at the molecular genetics. And indeed, we find that modulatory neurotransmitter systems seem to be involved in the genetic determination of these individually specific properties of the brain. And I'm happy to talk more about that if you're interested. So I'm trying to transition now to other timescales. So this is, let's see if I can get this started. This is the view in the fall out of my office at the Beckman Institute. And you can see that when the wind blows, the light and subtle parts of the tree, the leaves and the subtle branches vibrate quite quickly, and then the bigger and bigger parts of the tree move less and less rapidly with the same amount of wind force. So Rita Hari has a really nice paper on this topic about how our cognitive processes by nature are multi-timescale, because the statistical properties of the environment are multi-timescale. So this is Newton's second law, in which force equals mass times acceleration, which, given a certain fixed wind force, generates this inverse relationship between the movement, in this case, and the spatial frequency, from very small to very thick. So it's essentially a property of nature that we are processing multiple timescales at the same time. And this can be even better appreciated when we look at the auditory data modality, or sensory modality, rather. This is the pressure wave of a small part of a nursery rhyme. You might be familiar with it, but I spare you trying to sing it. And so the investigators here are showing amplitude modulation of the phonemes, of the syllables, and of the prosodic stress points, which are shown here as the yellow points. And you see that they are operating at quite different speeds, frequency ranges, and they're all part of the same signal, each of which needs to be processed concurrently in the brain, but by different distant sets of brain regions. And all of this is now embedded in slower time scales that we're processing, such as sentence level syntax and semantics, and that's embedded perhaps in paragraph level memory and larger semantic context. Much of these processes, by nature of their rapid timescales, are going to be missed by fMRI. And so this brings me to the fast connectome. And an approach that we're commonly taking in our lab is using scalp EEG. This applies also to MEG, so non-invasive almost whole brain coverage of real-time recordings. So let me walk you through this, which has really gained some speed over the last decade. So EEG signal at the channel level is pre-processed. As usual, artifacts are assessed and removed. We may or may not want to filter in certain frequency bands. And then the sensor level signals are source localized into a dense mesh. And those source signals are then averaged across the regions of our atlas of interest. So now we have region-wise time series for which we can look at the phase of the signal or at the amplitude envelope. And based on that, between every pair of brain regions, we can estimate phase coupling or amplitude coupling, essentially generating connectomes similar to the ones that I showed you in fMRI, even though they are not quite as crisp typically as the fMRI connectome. And then all kinds of graph measures, of course, can be applied. Once you have the adjacency matrix, you can apply similar tools that we have been very familiar with in the fMRI world. Of course, a big issue that I can't leave out here is the issue of source leakage in EEG and in MEG, which can create false appearance of connectivity from signals that are actually coming from the same source, so that it might appear at the scalp level that, you know, two different brain regions are actually interacting, whereas the signal is coming from the same source. So source leakage needs to be corrected. And unfortunately, this correction is extremely conservative in that it also removes real neural zero lag interactions, but it's something we have to live with. That said, all these limitations aside, I hope I can convince you today that there is really interesting and reliable signals using even these limited methods, and it opens an entirely different view onto connectome dynamics. So here we have a large data set that was shared with us. This is the Minnesota twin data set. And again, just pointing out that this is a classical twin study. And so here I'm showing a much shorter period of the recording. Remember what I was showing you in fMRI was perhaps 2.5 minutes or something like that. This is a much shorter recording from EEG, but you can already see that the connectome is much more rapidly switching between recurrent states that we have estimated using similar methods. Again, from this, we can derive fractional occupancies, transition probabilities, and perhaps other temporal characteristics of connectome dynamics. Using really similar approaches, we again see that the genetically more related monozygotic twins compared to dizygotic twins and unrelated participants really show different amounts of similarity of fractional occupancy and transition probabilities. This is true for all different frequency bands and almost all the different combinations of frequency bands and characteristics, so fractional occupancy or transition probabilities that we have looked at. What's really interesting is that in various different frequency bands, we find the same amount of heritability as fMRI or stronger. So in case of the alpha band, which is a really strong SNR signal in EEG, we find over 60% of the variance in fractional occupancy and transition probability explained by the genetic effects. And again, if we compare or we look at the association of the heritable connectome states and cognition, now I'm showing you here different cognitive factors factorized from a large battery, we find that in this young adult population, executive functions seem to be associated with transition probabilities measured in resting state EEG. So on the EEG side, the variance in this association comes almost exclusively from transition probabilities. Very little is coming from fractional occupancies. So it seems that at least in this young population, the way the states are sequenced, the particular sequence is what's driving the association with cognitive abilities. Maybe I should also mention, we looked at a lot of spatial properties of the connectome states. And by that, I mean, if you look at the actual connectivity matrix of the connectome states, and you compare that across participants, we've done various, anything that has come to our mind in graph space, as well as in the actual underlying edgewise connectivity, we find no heritability, and we find no association with cognitive outcomes, which is really shocking to us in this population. So it really seems to be the temporal characteristics. How the brain is moving through connectome states is what is actually heritable, not the spatial arrangement of those states. And so now I'm coming to the perhaps more exciting part of the talk, where I'm trying to put this all together. So when we look at the connectome through the lens of multiple data modalities concurrently. So let me first recap the traditional view as we think about the relationship between the different data modalities. So when neurons are active in the cortex, and we're talking here about masses of neurons that are geometrically aligned and with synchronized activity, they generate electrical currents and perpendicular magnetic fields that we can non-invasively measure using EEG and MEG. This is the real-time signal, right? But as we know, this neural activity also creates metabolic demands. Blood flow increases to bring fresh blood with lots of oxygen. Oxygen extraction increases, and this generates, through neurovascular coupling, the hemodynamic signal that the fMRI the MRI system is picking up in the fMRI signal. So the traditional view is that when we record EEG and fMRI concurrently, we mostly see the same neural processes. It's just that fMRI is particularly good at picking up where the activation is happening, and EEG is particularly good at essentially showing us the time course of that activity. And I want to argue in the following that that may be an incomplete picture at best. And again, I urge you, if you haven't seen Rita Hari's talk from OHBM 2019, it's on YouTube, to go and check that out. She really lays that out in a very long, cohesive interpretation for why EEG, MEG, and fMRI might actually capture vastly non-overlapping neural processes. I'm not saying exclusively, and I'm certainly not saying that fMRI does not have an electrophysiological basis. But what I'm saying is that the signal preferentially captured by scalp EEG and also intracranial, I'm coming to that in a moment, is perhaps not the signal that's driving most of the fMRI connectomics. So at least as we're talking about large-scale spatial patterns and co-activations, they might have non-overlapping windows onto neural activity. So often what we do in the lab is looking at concurrent EEG and fMRI data. We average the fMRI signal, as I showed you in the beginning, over certain parcels, and we source reconstruct the EEG to the same parcels, and now we can have time-resolved connectomes in the fMRI and in the concurrent EEG in spatially comparable manner, and then see how much of the dynamics actually corresponds or how they relate to each other. But before I come to non-invasive concurrent EEG fMRI, I want to actually start with invasive data. And I do that because I want to first establish an important understanding without worrying too much about source leakage, and then I move on to scalp data. So this is incredibly rare human data. Intracranial EEG in humans is recorded in neurosurgical patients that need to be observed by the neurologist for a certain period of time. And during that time, we can run experiments with them. And by we, I mean these generous collaborators, Louis Lemieux and his colleagues at UCL. In this case, they recorded intracranial EEG concurrently with fMRI, which is very rare in human recordings. So the method that we applied here was previously developed for fMRI data, so unimodal data, and let me walk you through that. So in this case, recording time is running from 0 to 20 minutes on this axis as well as on the other axis. And what the color scheme here shows is not temporal correlations as we looked at before, but it's actually spatial correlations between the functional connectivity matrix at one time point and the same functional connectivity matrix at a later time point. So this matrix is essentially a self-similarity matrix. So the connectome organization at the beginning of the recording may occur to some degree at later time points of the same recording. So again, this is just fMRI, and the same data is sliding over each other. We took this kind of an approach to bimodal setting now. So this is intracranial EEG time running on this axis and concurrent fMRI running on this other axis. When I plot the connectome here, remember that this is only just one part of the connectome in each patient. So they don't have whole brain coverage, but they typically have large grids that go over multiple lobes, and certainly over a variety of functional regions. So every patient has their own sort of window into a part of the connectome. So what you're seeing on this plane is a spatial correlation between these two data modalities. Remember that the corresponding physically corresponding time would run on the diagonal here. And if we account for 5-6 seconds of hemodynamic delay, then maybe the corresponding time would be slightly off diagonal. But what you're seeing here in the blue color are the time points in which the spatial similarity between the connectome states and the two data modalities exceeded chance level after correcting for all the multiple comparisons in this plane. And so what's surprising here is that the spatially similar time point pairs are not more probable in corresponding physical time than non-corresponding. What that means is spatially similar states do occur in EEG and fMRI, but they don't seem to occur in a temporally aligned manner. They're temporally asynchronous. And so this was true no matter whether we looked at phase coupling or amplitude coupling in the EEG. And it's interesting that we actually tend to see it in all EEG frequency bands. Once we observed this, we also replicated that in healthy young adults using non-invasive EEG fMRI with whole brain coverage. And so the next slide is going to tell you why I think this is really important. I think it's nice to visualize it in terms of the neurocognitive network, just so we appreciate how this deviates perhaps with our default understanding when we think about connectome dynamics from an fMRI point of view alone. So here on the top left, I'm showing a binary mask of one of the neurocognitive networks. This is the visual network. And then here I'm showing over a few minutes the temporal correlation, or sorry, the spatial correlation over time. So this is the time series of spatial correlation between each data frame, each frame of the connectome, and its similarity to this binary map. So co-activations in the brain are spontaneously going to be becoming more similar to a visually dominated map and then less similar again. So this happens spontaneously in the fMRI and each frequency band of the EEG, intracranial EEG. In fact, actually, this is scalp data, pardon me, because we have whole brain coverage, so scalp EEG. So if we now make a random cutoff here, let's say 2 standard deviations above the mean, and then we plot the connectome from these time points, you see that, as you would expect, obviously this is recursive, as you would expect, the organization of the connectome in these time points is really dominated by strong co-activations within the visual network. So the visual network in this case was in the top left of the matrix. If we do this for the EEG bands, we see a similar pattern. It's noisier, but you can appreciate that connectivity is still higher within the visual network compared to the rest of the connectome. So this is the delta band, and this is the EEG alpha frequency band. Now let's take a look at another network. And so this is the binary map of the default mode network here. And we did the same spatial correlations. How similar is the connectome state at every given moment in time to this map, right? And if we look at the time points where this activation was particularly strong, as expected, we see very strong co-activations within the default mode network. So the default mode here was at the bottom of the matrix. You can see a spatially somewhat similar connectome map in the different EEG frequencies, again, dominated by pretty strong connectivity in this part of the connectome. Again, a little bit noisier, but we can see it. So what's important about this plot is if you pick one particular moment in physical time, okay, so this is the recording time, and you pick one moment here, the fMRI connectome might actually be in a connectome state dominated by this particular pattern, which has these 2 large communities and a lot of connectivity within the default mode network. But at the same moment in time, if you look at a different time scale of the connectome, you actually see an entirely different connectome state. And this is because the time series of the connectome states are temporally independent. At least there is no linear relationship that we have been able to find between the different time scales. So if the connectome here in delta band is in this state, the fMRI derived connectome is in an entirely different state, and then the other EEG frequency bands are yet in another different connectome state. So to dive into this further, we now go to a final study that I want to discuss, which is, again, concurrent scalp EEG fMRI in resting state. But now, instead of defining the states in a data-driven manner using clustering or hidden Markov models, we are, in this case, taking an anatomically informed approach. So I'll lay that out here. So these are the most commonly studied 7 canonical neurocognitive networks, and we combine them into binary maps. This is a similar approach that we took and then extended as introduced by Spons and Detzel here. So every column here is essentially a binary map that comprises either one of these networks set to 1 as an active and the other ones are set to 0 as inactive, or it's a combination of 2 of them, 3, 4, and so on. So we can get 126 possible combinations of these 7 in terms of binary maps. Okay, so it would look something like that. They're just color coded from more simple to more complex, as in more networks involved. And these are just examples of what those activation maps might look like. We consider these blueprints, to which we then map the moment by moment co-activation patterns over the 68 regions of our parcellation. So for the bolt signal, we just look at every time point, instantaneous bolt activations of these regions, and we simply do a winner-take-all approach. Which one of these 126 is going to be the best fitting? We do the same for the band-limited amplitudes of the concurrent EEG signal in source space for the same brain regions. So if we look at the alpha frequency band over a few seconds, it'll look like this. So I'm here just showing Pearson spatial correlations of those binary 126 maps to the EEG alpha band amplitude. So I'm just showing spatial correlations for the first 7, because if I show you 126 time series, you won't be able to see anything. But as you can appreciate, the spatial correlation spontaneously goes up and down for each one of these maps. And if we then do this winner-take-all approach and then simply color code every time frame to the best fitting spatial map, we get something like this. And then we do this for various different EEG frequency bands, and we do this for the concurrent fMRI as well. So you can already visually see that the faster and faster EEG frequency bands have faster and faster state switches compared to the slower time scales. And interestingly, if you look at the fMRI over these few seconds of recording, these are 2.8 seconds, fMRI is in this particular brain state, and it is not the case that the EEG connectome states are more likely to be in this state during this period of time than other states. Again, this points to being temporally independent in terms of the state sequences. So of course we quantified this because, you know, a winner-take-all approach does not tell us if these blueprints actually fit the data or not, right? So for that, we generated different null models, one in which we spatially scramble the neurocognitive networks, the other one we temporally phase scramble the underlying signal time series of the brain. Here are the null models. This is one EEG fMRI data set. This is another one. Entirely different scanner, different sequence, and this is a pretty large EEG data set. We just wanted to have more power, no fMRI data here, but again, just to be convincing ourselves that the EEG source-based data actually fit these blueprints, these recombinations of neurocognitive networks. Not great, but certainly better than chance. So it's a really robust finding here. And so one last point of data before I get to the final conclusion. When we look at the statistical properties that I mentioned earlier, fractional occupancies and transition probabilities, we see that the fractional occupancies of the 126 brain states are not equally distributed. Some states occur more often than others, right? They occupy more of the total recording. The distribution of fractional occupancies over the 126 states is very similar, very high correlation values between fMRI and the various EEG frequency bands. The same actually holds true when we look at the transition probability matrix. So the likelihood of any pairs of states transitioning into each other, again, that matrix is very non-randomly distributed, and those transition probabilities are, again, very similar at all the time scales at which we computed the connectome. Similarly, the dwell time, the duration spent in a given state once you're in that state until you transition to another state. So in summary, the temporal sequences are very similar across the different time scales of the connectome, but they occur at very different speeds. So if you would take a the connectome sequence from the gamma band and you would stretch it out a lot, it would look very similar to the fMRI connectome because it obeys similar statistical properties in terms of how the states are sequenced and how long they last, each of them. However, it's really much speedier, much faster in the EEG connectome than it is in the fMRI connectome. And by definition then, the fMRI-derived connectome and the EEG-derived connectome cannot really primarily be following the same neural property, right? It's not true that if we just HRF convolve these fast connectomes, we end up with what we see in the fMRI. That is not the case, okay? They are temporally independent. It's not the case, in other words, that fMRI simply temporally blurs what's captured in the EEG connectome. And so let me get to the final conclusions and the notion of multiplexing. So the way we think our data makes sense, at the moment to us at least, is that the functional connectome comprises various streams of dynamics. There are multiple ones. It's not that the fMRI is measuring rapid dynamics and just that just temporally blurs them. Just seems that fMRI and EEG preferentially capture different viewpoints on connectome dynamics. The states are spatially similar above chance, not to a great degree, but there is some small to moderate level of spatial similarity. But those states occur in temporally independent manner, so at different speeds, even though they're obeying similar statistical properties in terms of how they are sequenced. And so we think of these different connectome streams as different parallel streams of information processing. So if we go back to this picture, imagine the total state space of the connectome was finite, okay? So when we do k-means clustering, or if we do HMMs, or even this blueprint matching, we have the simplified view that you can, to some degree, explain all of connectome dynamics within a finite state of space of states. Now, within that space, there is spatial overlap between what EEG and fMRI are capturing, right? So we're sharing to some degree the same state space in these different speeds of the connectome. But look at what happens when fMRI is moving spontaneously through this connectome state. It will go from one arrangement, a connectome, maybe connectome arrangement dominated by these particular connections, to the next, to the next. But the faster frequencies are going to be at a different part of the state space, moving much faster through a different trajectory, temporally independently of fMRI. And so what that means is that if you pick one particular moment in physical time, okay, you may not have one connectome arrangement. You may actually have a variety of different sets of brain regions that are communicating with each other in a somewhat independent manner. And I mean somewhat because I haven't even gotten into cross-frequency coupling and various other cross-timescale nonlinear relationships. But at least from a simplified and linear point of view, I don't think the idea that the connectome is moving through one stream of dynamics is compatible with our observations. So I call this the connectome multiplex because the idea of multiplexing in telecommunication systems is really a nice analogy to understand what might be happening. So in telecommunication systems, we have various different channels of content that are multiplexed over a single transmission system by tapping into different speeds. So they're encoded at different frequencies so they can actually be transmitted over a single system and then decoded on the other side. And this could be a good analogy to understand the streams of the connectome. And so why do we need these various different timescales of connectome dynamics? This brings me back to the notion of the fact that by its very nature, cognition is multi-timescale in a parallel manner. And this is true if we look at the sensory environment and the signals we're dealing with and encoding, as well as higher order cognitive functions, such as language, memory, and so on. They're all operating concurrently at various different time scales by nature, and therefore it would make sense that the brain and the whole brain connectome are operating at multiple time scales. And so that's my final conclusion for today. I hope that kind of convinced you a little bit to embrace EEG connectomics to some degree and think about the brain from multiple viewpoints or multiple time scales at the same time. Thank you.
Thank you. Thank you very much.